The overall goal of this experiment is to identify the downstream signaling cascade involved in cells stimulated with mitochondrial-derived peptides.
This method can answer key questions in the mitochondrial-derived peptide field, such as what signaling pathways are involved and where this occurs.
The main advantage of this technique is that it is straightforward and relatively easy to perform.
We first had the idea for this method when we examined human-mediated ERK signaling, which gave us limited information on the downstream effects. The subcellular fractionation method gave us a better idea of the role of human-mediated signaling.
On the third day after plating and washing 2 × 10⁶ SHSY5Y or HEC-293 cells according to the text protocol, dissolve S14G humanin peptides in 0.2 micron filtered distilled water, and reconstitute them as a 1 millimolar stock solution. Aliquot the pre-warmed serum-containing or serum-free medium in a 15 milliliter conical tube and add room temperature humanin peptide stock solution to make a working concentration. Immediately replace the medium in the cells with 6 milliliters of the peptide solution and incubate the cells for the appropriate amount of time. Use 10 milliliters of ice-cold PBS to wash the cells twice. Then scrape the cells off the plate in 5 milliliters.